Far too often, treatments and policies are developed to address cancer, not the people living with cancer. Friends knows that centering patients, their needs, and their quality of life can inform better research and change the trajectory of science. We're looking to the future of cancer care, bringing patients and their advocates to the table, making them partners in leading the charge. Our work, advocacy, and expertise is driving groundbreaking legislation that gets patients the treatments and care they urgently need faster than ever. Clinical trials historically have been restricted to a small number of patients. We're broadening eligibility criteria in cancer clinical trials to make them more accessible and equitable. Diagnostic testing can be inconsistent and variable. We've brought all players to the table to make sure tests are accurate so that personalized medicine can truly be precise. Too little information was being used from real-world patient experience to inform treatments. We're creating a new paradigm to shape how therapies are developed. And now we're charting a path for the next generation of breakthroughs. Breakthroughs like our CT Monitor project is one of the largest scale scientific endeavors in Friends history to assess the ability of a rapid and easy-to-use blood test to monitor treatment response. We're setting the standard for a new biomarker for use in immunotherapy, which will support informed decision-making for patients. We are transforming lives, and we'll use these 25 years of momentum to drive even more life-changing breakthroughs for years to come.
Good afternoon. I'm Ellen Sigal, Chair of Friends of Cancer Research. I want to thank you for joining us today for this important discussion on supporting development of diagnostic tests for unmet needs. This forum will feature a new report describing recommended strategies and policy considerations to optimize diagnostic test development for rare populations. This is important and a compelling need. Friends has and continues to be deeply invested in this important issue and the legislation supporting it. Critical to the future of diagnostic testing and personalized medicine is the VALID Act, which pushes for diagnostic reforms. They are much needed. Patients are our North Star in all we do. And this legislation would ensure patients receive accurate and reliable test results. There has been considerable interest and momentum of late in Congress surrounding VALID. That's why we're lucky to have with us two heroes in Congress, Representative Diana DeGette and Representative Larry Bucshon. They will discuss how they are prioritizing diagnostics in the current congressional session and some legislative solutions they are preparing. I am now honored to turn things over to Representative DeGette and Bucshon.
Thank you so much to Ellen and to Friends of Cancer Research for hosting this real important event. If there's one thing that we learned during COVID-19, it's the importance of having accurate and reliable diagnostic testing. Every day, healthcare providers and patients across the country use and rely on diagnostic tests to make important and often difficult decisions about their health. The accuracy and the reliability of these tests can literally mean the difference between life and death for some patients. For many years, public health officials have been calling for greater oversight of these tools to ensure the information that patients and their healthcare providers are relying on is as accurate as possible. And so that's why now, for almost a decade, Representative Bucshon and I have been working together on legislation to do exactly that. The VALID Act, which we introduced again last summer with Senators Bennett and Burr, would establish a framework within the FDA to regulate diagnostic tests, and it would give the agency the tools necessary to ensure that those tests are both safe and effective. Since introducing the bill, we've continued to engage with many of you, as well as FDA, to get your feedback and comments to ensure that we get this legislation right. And we've had tremendous success in developing breakthrough designation programs for medical devices and drugs at the FDA, We're looking forward to building on that progress with this bill by establishing a similar program that will allow for expedited review of diagnostic tests that addresses an unmet need for patients like people who have rare diseases. The framework that would be created under the VALID Act would provide a new way to approve diagnostic tests that seek to identify rare and infectious diseases like COVID-19 and countless other conditions, while at the same time ensuring patient safety. We can't thank all of you enough for your continued input and support of the legislation, and we look forward to continuing with all of you to work on the momentum that we've created and push this bill forward to the finish line. And with that, I want to turn it over to my co-conspirator, Mr. Bucshon, for his comments. Larry?
Well, thank you, my friend, Representative DeGette, for giving a great overview of all the hard work that we've done together over all these years on the VALID Act. And I also want to thank everyone at the Friends of Cancer Research for having us today and for continuing to be a valued partner in this important legislative effort. What drove me to start working on VALID was my experience as a doctor before coming to Congress. Healthcare providers and patients routinely use and increasingly rely on diagnostic tests to make difficult decisions about the best course of care and treatment. Unfortunately, we continue to see examples of some tests that don't meet the level of analytical and clinical accuracy that are needed to make reliable medical decisions, causing some patients to go through life-changing procedures that may not have been necessary. Congress must provide certainty, and that is what we are trying to accomplish through VALID. To provide certainty for patients that the results of their test are clinically accurate, provide certainty for doctors that the tests they are administering and making major healthcare decisions on are accurate. And lastly, to provide certainty for test developers and labs that the regulatory framework won't suddenly change and that they will have a clear understanding of what is expected from them within the risk-based framework. The sponsors have been mindful throughout this process to make sure that we are balancing patient safety while promoting innovation. For example, VALID provides certain flexibilities to help facilitate development and support innovation for diagnostic tests for rare patient populations, all while keeping in place high standards for patient safety. This is instrumental as we continue to move forward towards the future of precision medicine. With the help of the VALID Act, the future of precision medicine will enable physicians to provide more individualized patient care to discover, cure, and treat diseases that were previously unknown or untreatable. As for the next steps, Representative DeGette and I continue to work with stakeholders, the FDA, and committee leadership on a path forward. I'm working all the angles, as is Diana, to call on the committee for hearings on the VALID Act and advance the bill, whether on its own or attached to must-pass bills like MDUFA, which is coming up this year, which, as you all know, we're still waiting to receive from the FDA and the industry. As for in the Senate, it's my understanding that they are continuing to gather feedback from stakeholders in a bipartisan manner in hopes of advancing VALID. The time for Congress to clarify the rules of the road for diagnostic testing is now. I really think this year is a pivotal year for VALID, and I look forward to continuing to work with you all to help get it across the finish line so that we can provide certainty to patients and modernize regulatory oversight of in vitro diagnostic tests to better promote innovation advancements. Thank you very much again, and I'll turn it back over to my colleague, Congresswoman DeGette.
Thank you so much, and we'll turn it back over to the sponsors for the rest of this wonderful program.
Thank you very much, Representatives DeGette and Bucshon, for joining us today, and thank you for your continued efforts with the VALID Act. We look forward to continuing to work closely with you and your colleagues as the bill proceeds. I'd now like to introduce our main session for today's meeting. Over the past several months, we've been fortunate to work with a group of expert stakeholders to construct the white paper that you received earlier. The goal of this project is to examine opportunities to improve patient access to clinical trials and diagnostic tests and explore flexibilities to expedite the development of companion diagnostics, focusing on specific challenges with rare variants. To lead the discussion today, I'd like to hand things over to Megan Doyle, the Director of Global Regulatory and R&D Policy at Amgen, who will moderate this session. Megan?
Thank you so much, Jeff, and to Friends for convening this expert group and this webinar on such an important topic. As Jeff mentioned, we're here today to discuss the recommendations in the white paper, which hopefully you've all read, and which are geared toward expediting development of diagnostics for therapies that target rare biomarkers or indications. Facilitating the coordinated review and development of targeted therapies and companion diagnostics for these rare populations can be really challenging. And so to that end, the white paper outlines proposals to accomplish two goals. Number one, improve patient access to clinical trials of biomarker-driven investigational products for rare diseases. And number two, expand and expedite the development of companion diagnostic tests for these rare populations. So we'll discuss both of these in turn today with our panel. I'm Megan Doyle, and as Jeff mentioned, I'm with Amgen. And today on our panel, we also have with us Soma Ghosh from the Centers for Devices and Radiological Health at the US FDA, Joe Lennerz from Massachusetts General Hospital, Rasika Kalamegham from Genentech, a member of the Roche group, and last but certainly not least, Elizabeth Mansfield from Foundation Medicine, who will kick us off by walking us through some of the key points and recommendations from the paper. So I'll let you take it from here, Liz.
Thanks, Megan. And I want to thank Friends for bringing this group together and inviting us to this wonderful webinar. So I'm going to touch on some of the high points from the paper just as a quick review to ground our discussion. So we'll start with the idealized co-development model that was put forward a little over 10 years ago now, where a drug and a diagnostic are developed together, ultimately leading to an approved drug and an approved companion diagnostic. As many of us know, in the real world, it doesn't always work quite the way this diagram depicts, especially as we've found over the years where the biomarker or the indication is rare. As cancer therapies are being developed, more and more of them are now targeted towards very small populations, rare biomarkers. In order to enter into a clinical trial for such a drug now, patients are often tested with a variety of tests. And these tests may have differing performance characteristics depending on how they were developed. This can complicate not only the drug development due to potentially having a non-uniform population, but also when there's a separately developed companion diagnostic to be approved, the underlying varying performance for the enrollment assays may make the CDX performance appear worse than it really is. This work group got together and thought about how development for rare biomarkers or indications could be improved and still allow the process to provide quick and flexible therapeutic and diagnostic approval and within existing regulations and policies, and that we don't think there would be any need for a change in the law to do this. Next slide, please. Whoa, all over the place here. We proposed several regulatory flexibilities in this paper that we believe could help assure that safe and effective drugs and companion diagnostics could be developed and brought forward for patients. First of all, there are flexibilities that are already in place for drug developers, and these could be extended to companion diagnostic developers as well for rare indications. We think that the FDA could consider defining rare in the case of a companion diagnostic as 10,000 patients likely to have a particular diagnostic biomarker or indication. This is similar to the humanitarian device exemption number that has been proposed for VALID. However, often for a rare test, you have to screen a lot of people to get a few patients. But we would propose considering as rare 10,000 patients actually diagnosed. We propose implementing a modification to the traditional risk-benefit calculus to account for altered risk acceptability for rare situations. That would allow where patients are willing to take on more risk in order to gain access to therapies. And because FDA does have to publish their decisions and they are setting precedent, we propose that FDA could publish the risk-benefit assessment in the final summary of safety and effectiveness documentation for a PMA for a companion diagnostic, that it would explain that this precedent was only applicable in the setting of a rare biomarker or a rare indication, and that it wouldn't be generally applicable. There are a number of examples on this slide of where biomarkers may be very rare and where development of the companion diagnostic might be difficult. The working group believes that these revised frameworks could provide acceptable flexibility that would still offer a safe and effective drug and companion diagnostic, but speed up development and make it more flexible. Next slide, please. Other flexibilities that we believe would be beneficial for the development of rare drug companion diagnostic pairs are that FDA would allow companion diagnostic developers for analytical validation to rely on pre-existing platform level validation instead of always requiring intensive biomarker specific validation. And consider that similar types of mutations in the same genomic context will behave similarly. This would relieve the need to use precious clinical samples with rare biomarkers or from rare indications to perform the analytical validation. These are often difficult to find, and we believe that it may not be an appropriate use of such precious clinical samples. For clinical validation, we would recommend that FDA rely on the companion diagnostic clinical performance as assessed against clinical trial outcome, rather than requiring concordance with the clinical trial assay or LDTs. This is truly the measure of how well a companion diagnostic predicts an outcome. And if bridging to the clinical trial assay or LDTs is desired or required for some reason, that perhaps be moved to the post-market setting. We'd also like to propose allowing for pre-specified modifications for already approved companion diagnostic platforms that are seeking additional indications where the general platform has already been approved. This could considerably speed up the companion diagnostic review and approval timeline. We also propose allowing for post-market collection of real-world data and real-world evidence, something that I know that FDA is already targeting. In addition, do we have one more slide? No. I'm sorry, I can't see this. Here we go. We would like for FDA to consider allowing alternative samples for analytical validation, because as I mentioned before, these samples tend to be very difficult to come by and tend to hold up validation while diagnostic companies or laboratories search for them. These kind of samples could include contrived samples that are either engineered to have the particular mutation in question, or our natural cell lines, or some other methodology in which the mutations of interest are represented rather than using clinical samples. We'd also like FDA to consider allowing the use of similar tumors or tissues with the concept that DNA is DNA and that the tissue or the tumor type that it comes from is irrelevant, as long as the biomarker of interest is present. We propose the use of representative validation, and this means that for a platform like an NGS platform, the maker would demonstrate that different types of mutations, such as short variants and rearrangements in copy numbers, are adequately detected in various genomic contexts without necessarily focusing on the exact biomarker if samples are particularly rare. We'd also like to propose that FDA look at prior data of adequate analytical validation for the use of the assay, so that analytical validation is not performed over and over and over again on the same platform, where it may have already been established that, for example, interference or sensitivity are adequate. And so I'd like to thank, I've just done a quick run over of this, but I would like to thank the working group from multiple different sectors, including laboratories, diagnostic developers, and biopharma, for working together to come up with this proposal. And I hope we will have a fruitful discussion following this, and I'll hand it back to Megan.
Thank you so much, Liz. That was a really helpful overview and to ground all of us. So now that we're all on the same page, I'd like to discuss the points from the paper in more detail with our full panel. So, just to start off with let's start by discussing the current state of drug and diagnostic development for rare patient populations. And, I can say from my perspective, I would argue, as Liz kind of alluded to, the current paradigm tries to follow the approach we just saw in Liz's slide, but there are some key differences for co-development programs targeting these rare populations with high unmet need. And I would say first, the timeline's often quite compressed, particularly if you have a breakthrough therapy or a program that's eligible for accelerated approval or both. And second, that sponsors often have to go where the patients are, as the biomarker and the condition is really rare, which can impact, as you kind of alluded to, Liz, the tests and test results that are relied upon to enroll the early stage trials. So that's kind of my general overview of the current state. But Liz, from your perspective on the diagnostic manufacturer side at Foundation, what are the key challenges that you all see with co-developing a companion diagnostic for one of these rare biomarkers?
Thanks, Megan. I think it's most likely difficulty in procuring samples for analytical validation. There is a lot of competition for the same samples in the world. And when they start out rare, adding in competition makes it even more difficult. And secondly, we have seen in our development of companion diagnostics, when a number of tests are used to enroll patients into the trial, especially when they are local tests, all the tests are not always exceptionally well validated for the biomarker of interest. And so when we look at concordance with those tests, we see highly variable concordance depending on the tests. And it results in our performance looking not as great as it could in terms of concordance. However, when we look at clinical performance, our test tends to look very good. So it's primarily procuring samples and trying to compare to the enrollment test.
Got it. Okay. And Rasika, on the biopharma or drug sponsor side, how, from your perspective, is this impacting the way trials are being conducted for the drug and the diagnostic?
Yeah, thanks, Megan. It does have a pretty significant impact. I think Liz and you have already outlined some of those, the compressed timelines, this pressure to get the drug and the diagnostic out at the same time in this synchronous manner. And there are operational hurdles. Liz referred to the fact that sometimes the assays that are used are local assays. They're not the central assay. So we're often doubling back to figure out if that biomarker really is present in that sample. There are these trade-offs we have to constantly make with these precious samples, because Liz is right, they are clinical grade samples that are incredibly precious. But I would say the biggest impact of all is on patients. These are patients with rare cancers, with rare biomarkers. Often they do not have any other option for treatment. They've signed up to be on a clinical trial and they're facing all these roadblocks. They've been told at their academic medical center or wherever they got their screen done that they're eligible for a study, only to be told, hang on a second, we can't rely on whatever that was, whatever assay was used. We need to rebiopsy you. We need to redo the test. We need to figure out if you're eligible for this study. I think that is atrocious and not acceptable at all. And so that is the biggest impact. You know, these patients need a better option to get on study, and that's why we are here. That's why we as I, as a representative of biopharma, am sitting here, because you could just say this is a diagnostic problem. This is a test development problem. No, it's not. It is also a drug development problem. So hopefully that's what we're going to get to with some solutions and recommendations.
Thanks, Rasika. And Soma, given all of this, what steps has FDA taken to address some of these sort of challenges we're talking about in this space?
Thanks for the question, Megan, and thanks to all of you for inviting me to the discussion, especially because for most of the projects, I think FDA is part of the working group for this. FDA was not part of the working group that developed the white paper, so I think this discussion is very helpful for us to take back what the discussion is here and to discuss internally and for further dialogue with you in the future. So going back to the points that Liz had alluded to and to the points that Rasika described, I just want to start by saying that FDA acknowledges these challenges, and FDA is aware of these roadblocks and challenges because they are in constant communication with drug and device sponsors, even for development of these rare biomarker targeted therapies or devices. And I definitely would like to summarize a few steps that FDA has taken over the last few years to address some of these challenges. And I was glad when I was hearing to both of you, I was glad to kind of think that, yes, we are aligned on many of these steps that you're already proposing. So I would like to start by saying, many of you know that our review teams actually, they employ very flexible and least burdensome approaches when reviewing marketing applications. And this was no exception for PMAs for rare biomarkers. I would particularly like to highlight the example of the approval of Foundation 1 CDX for Entrec, because it's a very rare biomarker and it's one of the examples in the white paper. So for this particular review, we did take into consideration the rarity of the biomarker, and the review team leveraged the existing platform level validation data for gene fusions for all studies except the key studies, that is accuracy, precision, sensitivity, and specificity, similar to what has been proposed in the paper. Basically there were some remaining questions, but we took the totality of the data that was already available and the key analytical studies and the clinical validation study. And the review team determined that this was adequate for safety and effectiveness of the product for approval. And therefore, some of the remaining questions were actually shifted to the post-market setting, wherein we developed conditions of approval with the sponsor to be addressed in a timely manner. So we have, taken up different flexibilities and employed different level of flexibilities in our reviews across the board. I also wanted to refer to the drug approvals for rare populations, where FDA's current policy is to, based on the benefit risk calculus and the data that's reviewed, FDA's policy is to go ahead and approve the drug for rare populations, for populations with unmet need, without requiring the approval of a corresponding diagnostic product at the same time. And in these scenarios, we realize and recognize that these are not ideal for co-development. However, this has been our current policy in several instances. And currently FDA is themselves is considering and discussing alternative options to address some of the challenges in the current paradigm. The other example that I want to give is the finalization of the guidance document that refers to broadening the labeling of companion diagnostic products using companion diagnostic products for therapeutic products that belong to a group. And we actually rolled out this guidance and finalized this guidance, keeping in mind that it would better make the therapeutic options more flexible, and it would also alleviate the burden on the side of the drug and device sponsor to bring in another PMA when there are a couple of existing PMAs for some clinical validation. So across the board, we've assessed the existing policies, and depending on feedback, we've updated them whenever possible. And we are definitely open to future dialogue to work with you to further, broaden these different flexibilities. And to Rasika's point, I think Rasika was talking about very important, critical, points about co-development. And I just want to state that both CDRH and CDER have been open to patient enrollment based on multiple local tests. I think the expectation here is that the pharma should be able to determine what tests are high-performing tests and to have some level of comparability between the platforms so that the same group of patients are actually receiving the drug in the trial. So definitely I'll stop here and we'll continue the discussion, but I think this is extremely important conversation and FDA has been thinking along the same lines as all of you.
Thank you, Soma. That's really good to hear. And I think we all look forward to continuing this conversation with you and FDA as we move on after this webinar. So let's now move on in our panel discussion to appropriate safeguards and flexibilities. We kind of got into this a little bit with Liz's overview, but drilling down a little bit deeper, for tests being used to enroll patients onto a clinical trial, what types of data should labs make available to the study sponsor? Joe, maybe you could take that question first, and then Soma, I'd love to hear your perspective as well.
Certainly, thanks. I think the first part to keep in mind, as Soma just outlined, when trying to enroll a patient in a trial in a local laboratory or regional setting, there's typically something referred to as local testing or pre-screening for eligibility. So for a sponsor to decide whether a local test would be eligible for such an enrollment, they have to know some basic characteristics, or you could call that performance requirements. Now, as we've outlined in this white paper, those don't have to be extensive. It is the key performance characteristics such as precision and accuracy, sensitivity and specificity, but potentially also including some of those numbers, especially for rare biomarkers, as we have shown in the tables in the paper. It is those basic performance characteristics that would enable two things. First, of course, which is the premise of this entire work group, is access. Meaning, if there's a local test available that would be used for pre-screening, the sponsor should be available or should be made available the test performance characteristics, and the patients would have access to pre-screening or even primary eligibility testing. The second part, which I think is very important for meaningful implementation of a distributed enrollment strategy, is that this would give the sponsor the ability to harmonize across the different tests. So if one particular test have a completely different performance characteristic spectrum or a different approach for validation, for example, no availability of validation samples, like Liz has said earlier, those are really the rare and the key hurdles to make this available. I think that is some of the features that we outlined here to make testing as accurate as possible and as reliable. So the key performance characteristics that labs should provide are well known, and any credible lab assesses those during the validation. And so I believe that that is a meaningful way to drive adoption and access towards trials for cancer patients.
Yeah, I totally agree with you, Joe. And, just building on what I said before, I think I totally agree that there has to be minimum performance characteristics that are set and that are demonstrated by these different platforms. At the end of the day, the platforms should be comparable to each other so that they are enrolling the same type of intent to treat population in the trial. I think the platforms should have some minimum level of platform level validation also for the different markers that they can, that they are able to detect. And then for biomarker specific, you should, you've mentioned the performance characteristics they should demonstrate. Now that said, this is not a platform for me to come to a formal agreement. I think for that, I would encourage FOCR to submit a pre-submission to CDRH so that we can get more feedback, we can provide more feedback in a formal manner. But one thing I would like to add here is that for pharma partners, I think it is very critical to predefine the biomarker. I think the biomarker classification rules or the biomarker calling rules should be predefined and pre-specified such that all the enrolling sites and the enrolling tests use the same biomarker definition. That would really harmonize across the board before initiation of the trial, and we would not, as a regulatory agency, try to reconcile biomarker calling rules at the time of approval. So that's something I would definitely request the pharma partners and the diagnostics partners to think through in advancing the co-development programs.
Thanks, Soma. So we have a few more questions I wanted to get through with our panel, but I want to make sure we leave enough time for Q&A. So let's maybe get to, again, digging a little bit deeper on flexibilities that we are proposing in the paper. I'm wondering, if Liz, maybe you could speak to any in particular that you think from the paper that we could really apply today to mitigate the current challenges we're experiencing. You did a great overview of them in general. I was wondering if you have any in particular that you want to call out and speak to, and then Soma, would love to get your perspective from the FDA on those flexibilities.
So I think some of the most important flexibilities here are around the analytical validation and the samples that are used. Because we have such difficulty in procuring samples for analytical validation, procuring clinical samples for analytical validation, it seems that FDA may today be able to accept more contrived samples, for example, that have been demonstrated to have the mutation of interest, and that at least certain test developers can demonstrate that DNA is DNA in their test, and it doesn't matter where it comes from, so that we have more opportunities to go to, for example, more common tumor types. If we're looking for an approval in, I don't know, prostate cancer, but we really have most of our samples with that mutation are in lung cancer, it would be very much easier for us to validate on lung cancer. In addition, there are a lot of many tests that are on the market for companion diagnostics now have been approved for a number of indications. And using the existing data and confidence from those already approved indications, I think in the setting of high unmet need might be appropriate.
Soma, do you have anything to add?
Just quickly, I think, as I said, we are open to more flexibilities and additional discussion with you. And I would definitely also encourage you to gather the evidence for more flexibility and present it to FDA for further consideration.
I agree.
Thank you.
That there needs to be evidence that the things that we've proposed are actually realistic and would lead to a safe and effective test, but I think that that can be done.
Thanks.
Excellent. And so this will be our last panel question, because I then want to move on to the Q&A from all the stakeholders who've joined us. Rasika, based on your experience, how could these flexibilities be applied today within the current regulatory frameworks that we already have?
Yeah, thank you for that, Megan. I think, going back to what Liz said in her opening remarks, all of the recommendations that we made in the white paper, they're quite granular, really. And they can be, but they're not asking for a change in the statutory bar for either approval of the drug or the diagnostic. I mean, that's one thing we were quite we were quite determined to make sure that we were not asking for a change in the evidentiary bar. What we are asking for is how do we meet this high evidentiary bar while keeping in mind the practical considerations that we are talking about small numbers of patients with difficult to obtain clinical grade samples. So that's where this concept of contrived samples comes in. That's where this concept of can you leverage prior analytic validation data comes in. How do we do not want to dilute the bar for safety and efficacy. We do not want to dilute the bar for safe companion diagnostics at all, not at all. We do want to find a way to practically meet those bars. And that's what all of our recommendations speak to. So in terms of how FDA can do that, I think FDA has broad regulatory leeway and they can exercise flexibility. I think Soma spoke to quite a few of those, both on the drug side and the diagnostic side. What we'd like to see is perhaps something slightly more formal in the form of a guidance. I think there are lots of questions that people have as to when would these flexibilities kick in? How would you define something as being a rare cancer or a rare biomarker within, lung cancer? We understand that it's really hard to ask regulators to come up with absolute numbers, but if they give us any guidance, if they can give us any indication as to when they would consider these flexibilities to be applicable, I think that would be really helpful. The second thing that would be helpful is for them to take a look at the recommendations we've made in the white paper. And of course, as Liz said and Soma said, and we've all said, we're happy to continue the dialogue, but it would be helpful to know which of those recommendations are ones that resonate with the FDA, which ones do they think that they can implement today, and give all of us, the community, an indication of how we can progress from here. I think that would be great.
That's super helpful. Thanks, Rasika. I think that would be great. And so now moving on to our Q&A from all of our stakeholders, we thank all of you for joining us today and want to get to some of your questions. So our first question is from Ryan Hoshi at AbbVie. Thanks for joining us, Ryan. Ryan wanted to know if we can please clarify the definition of a rare biomarker versus a rare variant versus a rare disease. Joe, could you take that one for us?
Sure. So rare disease, as Liz had outlined, and I think it's currently proposed in ValidAct, and also we followed that in the white paper, is 10,000 patients being affected. Now, I think the difficulty when it comes to rare variant and rare biomarker, classically a rare variant is occurring in 1% of the reference population. Now, what that reference population is depends, of course, on the disease, so that can go down very fast. Now, if you look at, certain diseases occurring, for example, only in men or only in women, that may be a different reference population. Now, when it comes to within that subpopulation, a biomarker, for example, a predictive biomarker, illustrating a signature of that subset of the disease that would respond to a certain drug, we're talking about smaller and smaller pieces of the relevant population. About 10 years ago, that would have been probably at around the 10% mark. So a subset of patients would have to be at least 10% of the reference population for it to be a relevant biomarker. However, after identification of ALK and ROS, that number dropped down first to 3 to 5%, then to 1%, and I think with the recent NTRK inhibitors, I think we're at the 0.3% range right now. So in other words, rare biomarkers can be a smallest fraction of the disease, while variants are in the single percentage range and rare diseases in the 10,000 affected individuals. Now, what I think precision medicine is asking for, and that's why I really like this question, is are we defining lung cancer as the NTRK rearranged lung cancer as a new disease? So there is a precedent, for example, in the entire brain tumor field. The molecular signature has become part of the disease and name definition. And if this would be followed throughout, we're talking effectively about reducing many of the diseases to very, very narrow molecular subsets that receive a very targetable and hopefully meaningful and effective therapy. So these definitions are, of course, in flux. The percentages that I provided are in the white paper, but it's an excellent question. And so for now, I think we're talking about very small subsets.
Can I
Yeah.
jump on? Go ahead, Liz.
I was going to say, I agree. For the purposes of this particular white paper, I think we're just talking about very small populations. Whether that's defined by the exact type of tumor they have, whether that's defined by the biomarker stratification in a more common kinds of cancer, just anything where there are very few people affected.
Yeah, and going back to what I was saying about how do we meet these evidentiary bars while keeping in mind the practicality of these small patient numbers, that's really what prompted this discussion. And I would say this is where it would be really helpful to get FDA's thinking on how would they view it. We don't want exact numbers. We know that that's impossible, but what are maybe criteria where these flexibilities could kick in? I think that would be helpful.
I think we as industry could also help FDA understand at what point, at what population level does it become incredibly difficult to source samples to help set that bar if that's of interest.
Excellent. Thanks to all of you. So I know we have a number of questions. I'm going to try to get through a few in just a few minutes. So I'll ask everyone to be somewhat brief in their responses, and we'll try to get to as many questions as we can. We have a question from Janet Freeman Daly, a patient advocate, asking how can patient advocate groups for rare cancers help to increase available tissue for biomarker testing validation? I can say from my perspective, one thought I have on this is that we are seeing sort of, it's becoming increasingly more challenging to use leftover samples and banked samples due to increasing regulation, I think aimed at protecting patient privacy, of course, and patient rights to their own genetic material. But one thing I would say is that patient groups could speak up about how and when they're okay with their leftover samples and their banked samples being used. Of course, we want to make sure we have appropriate informed consent, but to the extent we make it harder to use those leftover samples and banked samples, then it makes analytical validation more challenging. I don't know if Liz or Joe, as the technicians on this panel, if you have any further thoughts on that.
I can take one quick comment. Is the FDA engages with a lot of multiple stakeholders on these issues. So one quick pointer is for any patient advocacy group, reach out to your FDA supported collaborative community. That's a specific term that the FDA uses to engage with multiple stakeholders. So find your area of interest and then reach out to that. Liz, handing it over to you.
Thanks, Joe. I really agree with Megan. The most important thing is to actually have access to as many samples as possible, and to the degree that patients can be encouraged to provide consent, broad consent for use. I think that would be incredibly helpful in making samples available.
Thanks to all of you. So now we have a question also from Allison Gannon, asking about, considering the difficulty in finding a suitably validated comparator test and to avoid those non-concordant results we talked about earlier in the panel, what do we as a group propose as a means of establishing performance? And I think we really addressed this in the white paper. So Joe, maybe you could speak to this, and or Liz.
Yeah, so I think we propose a minimum requirement.
Go ahead, Joe.
Sorry. I just get excited because there's actual numbers in the paper. Everyone was longing for that for so long, and now we have it. So we said 30 positive, 30 negative whenever possible. However, for some of the tumors, it's so incredibly rare that, we said 6 known positive with orthogonal confirmation will do. And for limit of detection, it can be 1 that we would dilute out. That's a proposal, of course, that's non-binding. But I think if we look at the rarity, that's sometimes the best we can do. So I cannot imagine that the FDA would require, 120 cases if they have never been reported. So I think there's a meaningful balance that we're trying to walk.
Can I quickly jump in here?
Go ahead.
Yeah, I just wanted to, again, reiterate that, for all our rare biomarker validation and approvals, we've required, not a very high number of positives ranging from 30 to 50 or not even 50 sometimes. And the negatives, we've asked for more because they are low prevalence, and to find one false positive, it's reasonable to screen a larger number of negatives. Just wanted to add that here. But that said, I think one more thing that FDA as a group has been requesting is generation of more standard materials and reference materials for both liquid biopsy and tissue biopsy tests, so that these materials, representing different variant types, can be used across platforms to compare the results of the platforms from one another, and also to establish and develop them. So that's one more thing that I wanted to add to this discussion. Thank you. Back to you, Megan.
Thank you, Soma. So I think this would be the last question we have time for from our stakeholders, and then we'll do our next steps discussion and send it over to Jeff. So from Daryl Pritchard at the Personalized Medicine Coalition, thanks for joining us, Daryl. Daryl is asking, how much does the FDA recognition of public tumor mutation databases, such as the MSK database, as validated sources of real-world clinical evidence, how much does that help in developing rare variant diagnostic tests? And can we expect more progress in recognized databases from other organizations than the FDA? I don't know if anyone, looks like Liz, you're nodding. Would you like to take that one? And then I'll kick it over to Soma.
Yeah, so thanks, Megan, and thanks for asking this question, Daryl. I think real-world data, real-world evidence can be enormously helpful. However, there has been a hurdle in connecting test results to clinical outcome for the purpose of companion diagnostics. And there are not a lot of databases that I'm aware of where a clinical outcome that is similar to a clinical trial outcome is actually recorded. So we may know the mutation, we may know something about the patient, how long they had progression-free survival may be a question. Is that actually recorded in real clinical life? We don't know. So I think that to some degree we need these databases, but we also need a reasonable discussion on how you assess the clinical outcome in the real world. It's not going to be by RECIST. It's not going to be by many of the measures that are used in a clinical trial. And so I think that's really the barrier that we need to overcome.
Yeah, I totally agree with you, Liz. And I would just like to add that we also advocate, FDA advocates getting results and supportive data from real world evidence. And, we are mostly encouraging that in the post-market space. And we have developed conditions of approval with our sponsors to supplement the pre-market data with more real-world evidence-based data. However, we do hear, as Liz was alluding to, that it's being a challenge because the endpoints are not clear. And just to point out, like when these protocols are developed, we have to discuss internally with other real-world evidence groups in FDA. And we are definitely open to discussing more of these strategies as to how they can serve as supportive supplemental data to the already existing clinical outcome data that we review generally.
Fantastic. So to wrap up our panel discussion, we just have a couple minutes. So I'm going to ask each person to maybe take 30 seconds to say, in your view, what are some specific next steps or incentives that could help drive change here and support the proposals we've outlined in the white paper? And I'll just go around the screen in the order that I see everyone. Maybe Soma could go first, and then Rasika, and then Liz, and then Joe.
Thanks, Megan. I think I would reiterate some of the points I've already made before, namely that we will continue this dialogue. We've already exercised a lot of flexibilities and we are open to exercising more. And I would also encourage FOCR to get the collaborators to generate some evidence to support some of the proposals that they are making in the paper. I mean, we would love to see whether, some of the evidences can be generated to show some of these complex variants are representative across the genome and across the panel. We would love to see that evidence and exercise more flexibilities. Thank you.
Thank you. And Rasika.
Thanks, Megan. First, I want to thank friends and everybody who has worked on this project for so long. I think one of the most important things we achieved here is bring together people from very, very different perspectives. We were able to speak with one another. And for me, a really good next step would be keeping the dialogue going. I take the point that Soma made that FDA was not a part of all of these discussions. I think that was a quirk of many things that were beyond our control, and we can certainly try to address that as we move forward. As a tangible next step, I think it does make sense for us to start with the recommendations laid out at the in the white paper, and start, dialoguing on how we can build on that. We're not asking, again, for statutory change or new rules. We're asking for flexibilities that can be exercised given our current regulatory framework. And we need to do this, as I always say, because our patients need it, and we cannot fail them.
Thanks, Rasika. Liz, and then Joe.
I think I can only concur with the two previous speakers. I would love to keep this dialogue moving along. I think that there are certain areas where we can perhaps present evidence very soon, and would love to have FDA take it under consideration.
Thanks. I second that, and I think this emphasizes the importance of regulatory science, where evidence should drive meaningful policy. Thank you.
Thank you, Joe. And I'll just say from my perspective, I agree with continuing the dialogue. I think it would be great to keep a group of thought leaders in the FDA together to have this discussion and talk about, can we develop other tools that help sponsors and regulators think about how and when to apply these flexibilities, even at a more granular level. I think that could be even really useful. And with that, I'd like to thank this panel for such a robust and interesting discussion. I know I've really enjoyed it and enjoyed being a part of this effort, and thank Friends for bringing us all together. And with that, I'll turn it over to Jeff for some closing remarks.
Great, thank you very much, Megan. I'd like to thank all of today's panelists and the entire working group that has shared their time and expertise to help work through the challenges that have been described today. As several of you noted, progress is being made, and hopefully collaborative efforts like this can help inform future actions and science-based policies. Thanks to all of you for joining today's meeting. Through our 2022 activities, we have a number of working group reports and research project data readouts for which we'll be hosting interactive venues like today's meeting. So to stay up to date on those and other important science and policy issues, you can scan the QR code at the bottom right of your screen and keep an eye out for more details in the coming weeks. Until then, take care. Thank you.